Hey, here we are, Advanced Anatomy and Physiology group. We're in the middle of week 3, thinking about the exams coming up at the end of this week. And once again, just a little reminder, if you're not watching these videos, I highly urge you to. And I know it's kind of funny to say that, because if you're not watching these videos, you're not hearing me urge you to say that. But there are things included in here that will help you and that will provide context. And I can take a look by views that a number of you are not watching these, and I publish these for you. I can't force you to. If you've got your own thing going and it's working, awesome. But I do feel like some people are missing some of the details, which are always available in the overview, but some people are missing the reminders that I give on a weekly basis. So for lecture this week, I've got a couple things to do. There's a detailed video on mitosis, and we're going to do a deep dive this week. Last week, we introduced the concept of mitosis and cell division and how the cell lives its life and why we want to have a division or need to have a divisional process that makes identical cells, which is mitosis, and have a divisional process that makes sperm cells and egg cells, which are haploid essentially. So there are some differences in addition to the similarities. So we've got mitosis video, the deep dive on that. Please be aware if it's in there, you're going to be potentially tested on those. The test this week and the questions on this will come from a large pool. Some of them are big picture, some of them are minute and detailed. But you treat these like a lecture that you can watch repeatedly. And also, hopefully you have closed caption on these. I just took a look at all of them. They should have closed caption. And that will definitely help. So notes are complete, and I know this is a little bit different than general anatomy and physiology. The programs to which you all are headed have asked us to do this because this is an example of many types of content delivery mechanisms that you'll see in the future. All right, so deep dive on the mitosis video, and then we'll have our first technical reading. And you're responsible for all the content in the reading, and it deals with cancer. Cancer essentially happens when mitosis, or the cell cycle, goes a little bit too rapidly. It talks about different kinds of cancer, talks about causes. And really, if you think about it like this, if there's a process in the body that happens at a given rate, like mitosis in a given tissue type for a given cell, problems can happen, or that cell can speed up for one of two reasons. The first reason is that the gas pedal gets pushed. There are things called positive cell cycle regulators that help move a cell through the cycle. If those processes, or if those enzymes, or if the DNA that control those gets messed up, essentially the gas pedal gets pushed on and the cell moves rapidly through the cycle and it divides and you end up with new cells sooner than you normally would. The other thing that can happen, and this is kind of cool, I think this is the subtle one, and it's amazing that they figured this stuff out. A cell also has negative cell cycle regulators, which are mechanisms that kind of keep the brakes on the cell to prevent it from going too fast. Cancer can also occur, in addition to being pushed on the gas pedal, cancer can also occur because the negative cell cycle regulators, or the processes that kind of keep things slow, get damaged. And sometimes it's due to environmental factors, sometimes it's genetic causes, sometimes it's inherited. So either if the positive cell cycle regulators get pushed too fast or the negative cell cycle regulators get removed, you can end up with a cell that goes through the cycle much more rapidly than it should, divides a little bit earlier, and then you end up with more cells than you should. If those cells have the ability to spread, or if they're able to metastasize, that's essentially cancer. So that's the lecture content. At the end of this week, we will not have a lecture quiz, you'll have a lecture exam. The lecture exam will contain 50 questions, and it will cover all the material from week 1 and week 2, and now this week, week 3. Please see the overview in Canvas, it lists those things. And I'm giving you this exam a little bit early because we'll have less lecture content on this first exam than we will on subsequent exams. I'd like you to have feedback. I'd like you to get off on the semester on a good foot in terms of grades. And there's a little bit less content on this exam than there will be on the subsequent exam. So that's the lecture exam. You can look at your old quizzes from week 1 and week 2. I've provided some sample problems that cover not only week 1 and 2 content, but also the content that we're learning this week on mitosis and cancer. And I would highly recommend going through and watching all the previous week's content delivery videos in order to prepare. You can't over prepare for this. You really can't. All right, for lab this week, we will talk about replication, which is the process by which DNA makes copies of itself. And you say, well, why the heck would DNA ever need to make copies of itself? Well, we know now, and we'll dial this in a little bit, that during the cell's life, there's a phase called the synthesis phase. And when a cell is in synthesis, it's kind of done living its normal life. It's gone through G1. It's done being a cell and having functions of whatever tissue it is and whatever cell type it is. And it's going to prepare for division. And in order to have daughter cells that are genetically identical, we've got to make copies of the DNA. A normal human cell has 46 pieces of DNA, and if we just split that cell, then you end up with 2 cells, each with 23. And we'll want to do that for egg cells and sperm cells, but now we're talking about making normal human body cells. So these autosomes have to be considered a little bit. So in order to make 2 cells, each with 46 for a while temporarily, and this will be during synthesis and beyond, that cell will have 92 pieces of DNA, so that when it divides or when cytokinesis occurs, each daughter cell has the appropriate amount. So in lab, we'll talk about the processes of replication, and there are some enzymes involved. So the way to tackle this is to first watch the generic video, it's not super generic, but sort of the overview video of replication, and then take a look at the written content in the lab that also deals with replication. So there are some enzymes, or there's some proteins that are key in facilitating certain events. So when you have a double-stranded piece of DNA, for starters, you have to unwind it. There's an enzyme that does that. Then you have to keep them apart. There's a mechanism for that. And then you have to start rebuilding and then seal it all back up. So the enzymes that are involved in replication are dealt with in the video and also in the technical reading. And it would be a great idea to be prepared for this exam if you could write out all the enzymes in the order in which they work. And then last but not least, in lab, there's a simulation that talks about the differences between DNA and RNA. And we haven't really dialed in RNA yet, but we'll talk about the functions of RNA. In general anatomy and physiology, it was just kind of briefly introduced. We said it helps to decode or interpret DNA. And this semester, when we talk about rough endoplasmic reticulum and ribosomes, and specifically when we talk about peptides and proteins, which we know are chains of amino acids, we have to ask ourselves, well, how are these amino acids assembled? There's a specific code. How does that code work? How do ribosomes know which amino acid to place in sequence? And really at the heart of it, DNA is essentially the recipe for those peptides and proteins. And RNA will be the facilitator that helps turn the recipe, the base recipe, into the actual product. All right, we've got a lab exam at the end of this week, and the lab exam will contain way less material than all of the subsequent lab exams because we've only had 2 weeks. Last week and this week. So last week we did some heredity content. Please go back through, take a look at Punnett squares and probability again. There'll be a few of those on the lab exam. And then this week we're going to deal with replication and DNA and RNA. Seems like a lot, I know. Thank you for the work. Hopefully Canvas is working for you. If you need me, you can always get in touch with me. But so for this week for lecture, we've got mitosis video and cancer reading. For lab, we've got replication video, replication worksheet, and some content in there. And then we've got a simulation that's got to be complete, and that simulation can be found in Connect. All of this is in preparation for 2 exams at the end of this week, and they will be from Sunday morning to Monday morning, like they always will be. I'll have to go through and hand grade each individual exam, so please hear me on this. When you push submit, and some number populates immediately after you've submitted your exam, please know that I have not yet then had a chance to go back and take a look at that. So that initial score that you see will not be your final score. It'll take me maybe a day or so to go through every question on both the lecture exam and the lab exam to make sure you get credit for the questions for which you deserve credit. And then I'll send everybody an email. I'll make those exams available so you can take a look at them. And then we can close the loop, and then we're going to be on to week 4 already. So this is supposed to be kind of a pep talk. I know I'm kind of awful at those, but please make sure you're studying. Please make sure you're aware of what's going on. Please make sure you're aware of the exam window. You've all signed the syllabus acknowledgement, so you know that you're governed by the policies contained within. If you need me, please reach out. Make sure you're checking your emails a couple times a day, and I hope you're watching these videos.